Hello, everybody. My name is George Gavrilidis, and I'm a postdoc researcher at INABS Earth in Thessaloniki, Greece. And I'm very fortunate to be able to talk to you about perturbation modeling in single-cell omics through the Elixir single-cell community, in which me and my collaborators are really active and really engaged into trying to understand complex single-cell biology with no, potential translational output. So the single-cell technologies are becoming more and more robust, and they are capturing nowadays many facets of the cellular physiology, like the transcriptomic side, the epigenetic side, or the metabolomic or the proteomic side. And also, we do have right now technologies that are actually try to understand the spatial organization of single-cell populations. We have the spatial multi-omics, which are actually trying to capture the microanatomical organization of tissues and organs. And of course, all these complex single cell technologies with significant throughput demand computational tools that are becoming more and more sophisticated, trying to actually accommodate the needs that exist for deciphering the biological complexity of these datasets, and also computational tools that are trying to use computational infrastructures with compute power to do very intricate modeling using machine learning and deep learning. From the typical single cell analysis that we have post the ordinary quality control preprocessing, we have various topics and various types of analysis like clustering or reference mapping or trajectory inference. But the focus of this presentation is on a specific type of analysis that has a strong translational footprint in terms of systems medicine, and that is perturbation modeling along with cell type compositional analysis. These are two terms that are used usually intertwined. I'm going to use the term perturbation modeling henceforth. So what is actually perturbation in single cell biology? This is a really interesting topic in our opinion, and the perturbation can be found in several scenarios. The first most common scenario is when we are analyzing samples of control cases and cases that bear some disease phenotype. So in typical control versus disease cases, the perturbation can be actually the onset of the disease and the pressure it places on control populations. A second, more straightforward perturbation scenario has to do with single cell experiments that come from CRISPR genetic manipulations or high throughput screenings with drugs and chemical compounds. This type of analysis provide the unparalleled opportunity to actually test and experiment with multiple perturbations or interventions at the same time, and try to glean some insights that could potentially be really useful for therapeutic purposes. The third scenario is a little bit more complicated, and it's like a mesh of things that we talked in so far. We're talking about multi-omic single-cell experiments that also contain some kind of perturbation, like a CRISPR or a drug perturbation. So these datasets are more complex, but for this reason, they are even more interesting in terms of therapeutic potential, because they can show us how the perturbation affects multiple omic modalities across a cellular ecosystem. The objectives of perturbation modeling have been discussed in the literature. They are being revised every couple of years, but the basic pillars are these 5 objectives that you see on this slide. Starting from the one that is the most easily tractable, the most easily comprehensible, the first objective is actually uh, the objective that has to do with elucidating perturbations from confounding sources of variation. This objective actually reflects the need to improve the signal-to-noise ratio that is a basic component of this of the datasets, the single-cell datasets that come from experiments with CRISPR manipulation or drug screenings. There are some cells that actually evade perturbation or experience perturbation to some degree. So this the perturbation modeling in these cases has to do about actually finding which populations are mostly affected by the perturbation and removing the other sources of variation. Meaning again cells that are not experiencing the perturbation at all, or they are experiencing a partial effect. The second objective has to do with the creation of a pharmacogenomic latent space in which we can categorize drugs based on their omic, based on their capacity to actually affect the single cell omics. And hence we can classify drugs and known compounds to pre-existing categories. And this whole approach actually tries to bridge the effect of the drugs and the changes on the omic level. And of course, we can actually extract some conclusions about the way that the drugs actually operate, the signaling pathways they affect, the targets, the molecular targets that they are actually focused on, etc., etc. So the third objective is somewhat similar. It's like a bridge between chemoinformatics and single-cell omics. It has to do about predictions in terms of chemical structures of chemical compounds, based on the omic information that we have from the single-cell experiments. And this is really, really interesting and important in terms of efforts that have to do with drug repurposing and drug repositioning. A fourth objective has to do with being able to infer perturbation synergies. In this type of objective, we're talking about models that are able to be trained on singular perturbations and then create predictions about potential synergies of these perturbations. And this is actually really important because in several therapeutic regimens in the clinical setting, we're very much interested um, in combining two or more pharmaceuticals, especially in aggressive cases where the patients are in urgent need of some alternative therapeutic protocol to change their dismal clinical course. And then we get to the last objective that has to do with extrapolation to unseen events. This is the most ambitious objective, the most ambitious of all. And we're focusing here on models that are being trained on specific single cell data, and they are trying to generalize to unseen events, meaning that they are trying to predict what would be the aftermath of a perturbation on some control cell types for which we don't actually have real experimental data from the wet lab. So if we know that that certain cell types change after perturbation, then we cannot actually predict about a cell group that we've never tested, how it would look like after a potential perturbation. And this has a number of advantages as an approach, and this is a really critical objective because we can actually start to characterize the otherwise infinite perturbation space, because it's not feasible always to test in a wet lab, in actual laboratories, uh, all the perturbations we want in all the cell types that we want to examine. Since now we have set up a framework about perturbation modeling, I think it's a good time to actually indicate and showcase this paper that we recently published, a mini review on perturbation modeling. And this paper is briefly addressing all these issues that were mentioned before, and then focuses mostly on the method side, on the tools that exist right now to actually do all this perturbation modeling on the various types of single cell technologies, single cell data sets. We're talking about 40 methods right now, and the number is growing rapidly. So by the end of the year, we are projecting around 50 or 60 tools. And um, we were a little bit puzzled on how to group these models. So we devised a way of clustering them and grouping them based on some features. And these features had to do with the algorithmic architecture, computational architecture of these tools, but also had to do with the perturbation modeling objectives that they are trying to address. Because we are lacking right now a model that fits all the molds. I mean, a model that can do that can address all the objectives of perturbation modeling. So to actually elaborate a little bit on these six clusters, we're talking about some shallow models in the beginning. These are some of the oldest models that we have since 5 years ago. Biostatistical models and simple machine learning models that are trying to do some modeling. The second group has to do with mixed machine learning models, models that blend and dovetail several machine learning um, uh, toolboxes and architectures. They are blending, for example, probabilistic learning with clustering. And of course, they're trying to discern cell states that are affected by the perturbation. And some of these models also are trying to uh, venture on some kind of a causal machine learning analysis of perturbation modeling, which is really interesting. And it will definitely see a lot of attention in the future. A third group has to do with models that are mostly interested in perturbation similarity, meaning that they are trying to focus on the pharmacogenomic latent space and try to see the drugs and the genes, how they are connected and how we can extrapolate from one to the other. Another group has to do with complex generative models. This is the biggest group of perturbation modeling tools that we have right now. And we are talking about deep learning architectures that have to do mostly with autoencoders, but not only that. And these models are definitely really capable of doing some extrapolation to unseen events. We're also seeing some models that exhibit some sort of biological explainability. And they are definitely the most, I would say, usual models in perturbation modeling. We have another group that has to do with gene regulatory networks. These models are actually trying to reverse engineer gene regulatory networks per cell group, and then they're trying to do some kind of digital knockout simulation on these GRNs. These are really potent models and really interesting. And since the need to actually simulate digital knockouts or knock-ins without having any prior knowledge is so this need is really present. We are expecting this particular group of tools to become even larger in the coming months or years. Last but definitely not least, we're having this group of tools that are taking the field by storm. And these tools are foundational models that are trying to emulate the example of large language models like ChatGPT. And these models are being trained on millions of cells and datasets like the Cell Atlas or Tabula Muris or Tabula Sapiens. And then they allow the user to fine-tune the overall architecture based on his or her own dataset or the needs that the user has. So these models then can perform a number of tasks, one of which is perturbation modeling. So these models are not perturbation modeling specific, but they have this type of analysis in their broader repertoire. On the right, you can actually see several four schemas that are trying to depict um, some of these architectures that we are discussing right now. So In this upper left part, we have a variational autoencoder that is trying to do the extrapolation to unseen events. And you can see here that some of these architectures are equipped with some sort of biological explainability. Another type of model has to do with the implementation of the optimal transport theorem, that is actually a reflection of causal machine learning. These efforts are focused on trying to understand the counterfactuals and the way that they're trying actually to recreate and map how cells change after the perturbation, what are their exact counterparts after the perturbation. And this is really important when we're trying to understand the cell states that change after a specific perturbation. Some cell states get vanished, some cell states emerge. The third schema has to do with the GRN prioritizing models. This is a schema that actually illustrates that these models can recreate GRNs, they can simulate digital knockouts, and they can predict the knockout responsive genes. So they allow the user to actually do simulations about a systemic perturbance across the whole system, a systemic perturbation across the whole cellular system within the single cell data space. And of course, here we have a depiction of how the foundational models are trying to tackle the perturbation modeling issue after being pre-trained on external databases or on a large swath of single cell data. They are capable of doing some perturbation modeling and providing some really interesting insights.